Hello, everyone. Thanks for tuning in. Today, I would like to speak to you about how to unlock single-cell discovery in formal and fixed paraffin-embedded tissues with the CODEX technology. My name is Oliver, and I'll be the host of this webinar. Cancer research and discovery has embraced single-cell biology. Now, by single-cell biology, we mean techniques like single-cell whole transcriptome sequencing or CyTOF, both of which are able to take a tissue, like breast cancer in this case, and break it down into its cellular constituents in a very detailed and a very nuanced manner. So this is accomplished by analyzing the expression patterns of hundreds or thousands of mRNAs or proteins. And the result from it is a very, very, very accurate overview of what types of cells are within this tissue and what state of cells these are in. In this case, you can see a T-SNE plot. A T-SNE plot is one such visualization that allows you to analyze these types or to understand these types of multiplex data. And in this type of visualization, each cell is a dot, and each color shows you what type of cell there is. So for example, you can see these blue dots. These blue dots are all individual cells, and they're all basal cells. The fewer dots you have, the fewer cells you have. The more you have, the more. So it gives you a really nice overview of what's going on inside of this tissue. This is an actual breakdown of a breast cancer tissue that we will speak about today. Now, as good as it is, this technique does have some drawbacks. It's not always compatible with FFPE tissue, and it generally comes at the loss of spatial resolution. At the other end of the spectrum is FFPE histopathology. So techniques like H&E staining or chromogenic immunohistochemistry are the gold standard for clinical analysis of patient tissues. But they've really not allow us to do highly multiplexed analysis of these tissues. Now you can see an example here of keratin-8 staining in breast cancer. You can see with great precision each individual cell, which means that you have single-cell spatial resolution. But the staining of keratin-8 is not that much different of the staining of keratin-18, which I've just toggled over to. And then, again, not that much different from keratin-19. So let me toggle through these three markers. Now that I've pointed it out to you, you can see the differences between the markers. But would you have noticed that if I wouldn't have told you? This is a general problem, and people generally recognize that if we use fluorescence microscopy, we see more. So what you can see here are the same three markers: keratin-8, which is blue in the image, but white here for better visualization, keratin-18 in red, and keratin-9 in green. You can more readily see differences, especially small differences, between these different types of breast tissue epithelia. There's an epithelium that labels primarily with keratin-19. Here is one that may have more keratin-18 than keratin-8. On the flip side, this one has more blue and thus keratin-8 than others. This type of imaging, fluorescence microscopy, has a higher dynamic range. But up to this point, or up to recently, people have mainly been operating with about 3 or 4 fluorophores. So the real challenge is to add more fluorophores, like beta-catenin, PCNA, to show you proliferating cells, or TP63 to show you basal cells in this epithelium. And once you're done with that, you want to put on more labels, like E2F1, MHC, different types of keratins and collagens. This is what we will speak about today. We're going to talk about the CODEX technology, which enables you to do single-cell biology by putting up to 40 or more biomarkers on an intact piece of tissue. So by virtue of that, CODEX gives you sort of an in-between space between single-cell biology techniques, like transcriptome sequencing, and FFPE histopathology, which gives you the single-cell resolution. CODEX gives you that single-cell resolution, and it gives you highly multiplexed biomarker imaging, which allows you to take a tissue at single-cell spatial resolution, break it down into its individual cellular constituents, and therefore produce unprecedented insights and analyses of these tissues. Now, I would like to emphasize here that this breast cancer tissue will be the topic of our discussion today, and this tissue and the subsequent analysis is also what has actually produced this T-SNE plot, this single-cell biology result. This was generated entirely with the software that comes with our CODEX system. Now I'll walk you through that. Prior to doing so, though, I just would like to show you the CODEX system. So this system was launched in 2019 after it was initially developed by Gary Nolan's lab at Stanford. We are very happy with this system because it is the first and only benchtop solution for spatially resolved ultra-high multiplexed immunofluorescence. This box is really quite small. It fits on any benchtop in any lab, and it integrates seamlessly with existing microscopy devices. Fluid handling steps and staining steps are automated, so the researcher must not or does not have to use his or her hands to do this experiment. And I will show you how this experiment occurs. So a researcher will develop an antibody panel with up to 40 antibodies. We have a catalog online. Researcher can choose the antibodies that they're interested in. Or they take antibodies that are not in our catalog and they conjugate them with oligos, which are important to run this experiment. The antibodies, the primary antibodies I should emphasize, are then all put onto a piece of tissue at once and then stained on the benchtop. I would like to say again, this is the only manual handling step that has to be done. After this point, the tissue with the antibodies on it goes into the CODEX machine, and the first reveal cycle begins. During the reveal cycle, up to three antibodies that are labeling the tissue and that are equipped with barcodes, unique barcodes, will be paired with reporter readout probes. So these are fluorescent molecules, red, green, blue, that are paired with an oligo. These oligos will bind to the barcode on the antibody and thus allow the microscope to take an image. That will be one cycle. After this cycle, the reporters are removed and the experiment repeats itself. New oligos with reporters are put on and another picture is taken. This is fully automated. The researcher does not have to do this. So once this process is continued or completed and 40 biomarkers are read out, the researcher gets a multiplexed image. This is an example of this. Here you can see a breast cancer, 36 antibodies that have been labeled. You can see the tissue, the whole tissue that was imaged. It was 1.9 by 3.56 millimeters in dimension. On the bottom here, you can see the antibodies that are color-coded, matching with what they are here, where P53, for example, will be shown in yellow, and Ki67, which is rather rare, but you can see some cells here, and up here, there are a couple of them. Everything is matched here. This is breast cancer tissue. So for those of you who don't know, there are certain types of epithelia in here, and they will be the topic of today's discussion. So what you can see here, for example, these are secretory epithelia. This big area. Each one of these round circles is this type of sort of a secretory epithelium. We call them also lobules. Now, these are the areas of the breast tissue that produce lactation fluids. And in this case, this is breast cancer tissue. I should emphasize this is a stage 2 ductal adenocarcinoma. These lobules are swollen or hyperplastic. You can see these big black spaces here in the middle. They're not supposed to be there. They look very different in normal tissue. Now, on the other end of the spectrum, you have a collecting duct. This is, I would say, a more rigid structure. It collects the fluids that are generated by your secretory epithelia, and is also not hyperplastic or swollen in this particular instance. Now, you can see I have 7 antibodies on this image. It is a very revealing image. It's aesthetically pleasing, and it's data rich. Now I'm toggled over to 5 other antibodies. This is the exact same tissue with an additional 5 antibodies. Here are 5 more. Here are 5 more. And I'm going to continue on until we've seen all 36 antibodies. And I would just like you to appreciate not only how beautiful this is, but how much data are contained in this image, in this series of images. Is overwhelming in some sense. And therefore, the question likely arises, how do we analyze these data? How do we interpret this? And that will be the topic of the next part of this seminar. So the first step in our data analysis is cell segmentation. So we are using a watershed algorithm based on the DAPI signal that breaks down each cell into a little segmented line. Resulting from this, every cell will have an identity, which is essentially a number. In this experiment, we segmented 63,056 cells. That is the total number on the image that I had just shown you. Every cell will then be labeled with up to 36 antibodies or not. So they will have unique readouts. And for example, this cell here, number 5, is labeled very, very weakly. So this is a 16-bit image range. It goes up to 50,000 or something. So this cell is probably not labeled. But then you'll have a cell like cell number 2, AB1, 48,000. So this number indicates that the cell is probably labeled quite nicely. This will be a unique pattern over 36 antibodies and for every one of these 63,000 cells. So you could call this very highly multiplexed data. Now, in contrast to some other single cell techniques or some other multiplexing techniques that we use or that people use, like single cell genomic sequencing, we have spatial coordinates. This is key. This is an in situ result. We know exactly where every cell is. So how do we analyze these data? Now, I'm not going to focus so much on these spatial coordinates, but much more on the multiplexing and the different types of cells and the cell phenotyping. And basically, our experiments revolves around the idea of unbiased clustering, where you start with a huge population of diverse cells, and then you rely on a computer algorithm to organize these cells into distinct clusters based on similarities in their biomarker expressions. So here you can see, this is just a demo version from Google AI. Different types of cell populations in different colors have been identified based on some type of similarity. In our case, this purple region here would be a cell type that labels very strongly with AB1, for example, very weakly with AB2. And all of the other dots in this purple region will be doing the same thing. This brown one, they will be labeling very strongly with AB36 and very weakly with AB1, for example. It's a combinatorial labeling pattern. It's very complex. That's why we rely on a computer. It makes sense for us. But the point more so, or the really, really important point here is this is unbiased. We're letting an algorithm decide for us what cells are there. Now, this can be good or bad. If you don't actually know what you're looking for, this could go out of control because a computer can come up with hundreds and hundreds of different cell types. So you should have some type of a preconceived or some type of a basic understanding of what is possible. But it's still unbiased, which is really good because discovery by its nature should be unbiased. You should let your results guide you. And this is what I appreciate most about this. I'm letting these data guide me. And I'm going to say right here, for full disclosure, I'm actually a neurobiologist. I was a neurobiologist prior to joining this company, and I'm presenting an analysis of breast cancer tissue. I had zero preconceived idea about this tissue. I didn't know what I was looking at. So everything that I will show you now are the data guiding me based on unbiased analysis. So what we see in the next tissue, these are MAV-generated T-SNE data. So MAV is the multi-analysis viewer that I have used to generate every data point that you see today. The multi-analysis viewer is the Akoya software for CODEX. And we generate T-SNE plots in here. What you can see here are all 63,000 cells from the breast cancer tissue clustered into distinct two-dimensional geographies. So you can see these little islands here. Each one of these is geographically distinct. And the blue and green sort of indicates the density of cells. So you can see there are a few cells here, but they're pretty closely packed, probably because they cluster very tightly with each other. Here you can see rather loose, meaning that the similarities are there, but it could be tighter clustering. This here shows you something that's more easier to understand, and those are the biomarker intensities. Each biomarker is distinctly represented across the geography of this T-SNE plot. You can see the biomarker name, and then it toggles through. Red means hot, or a lot of this biomarker. Blue means cold, or a little of this biomarker. And in this case, keratin 19 is very strongly expressed in these cells and very weakly expressed in these cells. Finally, this is the output that you get from this after you've analyzed data. So what I've done here is I've annotated different types of cells based on their biomarker expression. For example, the cells over here on the right side, labeling strongly with keratin 19 and many other biomarkers, as you will see, those are luminal cells. We know that. So the user annotates that and thereby produces these populations. Now, I will dwell on these two results to show you the power of the CODEX system, and especially the power of toggling back and forth between these these sort of clustered data and real morphological data. Let's start with this example. So this is the expression of keratin 14, and you can see it's very limited in a small population of cells over here. Now, let me also emphasize that this would be the end result of many single-cell sequencing studies, for example, because they don't have spatial resolution. They would, or we, if we did this experiment, would just know that there are these keratin 14 cells. We would know the number, we know they're distinct, and we probably know that this would change in a different experimental condition. But the lovely thing about CODEX, any type of spatial multiplexed analysis, is that we have side by side. We have these types of, let's call them big data, and we actually have the anatomical substrate where this came from. So here you can see in red keratin 14. This is a pseudo-color map where you can see keratin 14, strong expression. You can see the arbitrary units on the 16-bit scale, very, very strongly expressing in these ducts. You see there are not that many ducts here, which corresponds very nicely with there not being that many cells here. There are way, way more cells that have sort of an intermediate keratin 14 expression. You can see this all in yellow and green. These will be your secretory epithelia. Now, it's really, really important that this is actually reliable. So I would like to say a word about the dynamic range of the biomarker signals in a CODEX experiment. And what I'm showing you here is keratin 14, which is the same marker that we had just looked like. Go back, keratin 14. This is a pseudo-color map, but now we're showing it in red. And we're also showing keratin 8 in DAPI. So what I'm doing here is I'm taking a close-up view of this duct. You can see very strongly red line around here, which is your keratin 14. And on the inside of your duct and in an adjacent lobule, you have a secretory epithelium. Labels quite nicely with keratin 8, but not so much with keratin 14. So you can see there's quite a range of staining, especially in the red channel. You see this weak red over here and very strong red over here. Now, again, the clustering algorithm depends very much on this range. This biomarker range needs to be faithfully represented. So it is key for us to communicate to you here that this result is reliable. And what we've discovered as we were doing this analysis is that indeed, this result is a perfect replication of what authors have described previously in an unrelated study. Now, before I dive into this, I would just like to say this is an entirely unintentional replication of a result. We did this experiment, we designed the antibody panel, we even did most of the analysis prior to realizing that this paper in Nature Communications, which was published in 2018, exists. So what do you see here? Here you see a duct, very much like the duct that we see. And this duct has a lovely red ring around it. These are basal cells. You can see a high magnification overview here. Compare that to ours. They don't look that dissimilar. In fact, they almost look exactly the same. Here's a lobule. You can see the very weak staining with keratin 14 around it. Here's a lobule in our experiment. Once again, it's almost perfect recapitulation of this result. If you look at the histograms here, you can see the keratin 14 expression for basal and myoepithelia. You can see very strong expression of keratin 14, this red ring. If you look at luminal cells, you can see much weaker expression of this marker. This is our analysis from our tissue. It's almost exactly the same. You get about a fourfold difference, threefold difference between luminal and basal epithelia in the intensity of keratin 14 expression. That is this result. This result was done by Akoya, and it was done at UCI years before. We were not aware of this result, but we have perfectly recapitulated it. That being said, CODEX produces reliable staining over a range of biomarker expression intensities. This is a key point. This is very good to know. And it should give you, and it definitely gives us, some confidence that analyzing our data is both reliable and faithfully representative of what's happening in your biological tissue. So the dynamic range of biomarker signals that CODEX captures is very good, and it's very reliable. So now that I've said that, I would like you to appreciate this map of T-SNE plots. So you can see every biomarker, every one of the 36 that we apply to this tissue, and you can see they have very different expression patterns. Probably you could spend an hour looking at this map and writing up hypotheses and stories, but we don't have that much time. So we're going to focus just on secretory and non-secretory epithelia. So if you look over here on the left side, you can see very nicely this little cluster of cells that I briefly mentioned already. It labels strongly with keratin 17, 5, and 14. It's red, hot means strong labeling, red, hot. Cold, blue means very weak labeling. So you can see these markers are quite specific, but they do wash into the luminal space here, which is yellow and green. On the other side of the spectrum, you have this cyan squares here. You have keratin 19, keratin 18, and keratin 8. You can see they label very strongly on the right-hand side of the T-SNE plot, but not this little island here. So very distinct. This is essentially the opposite end of the spectrum. So we're going to look at these data now, and we're going to see how these epithelia differ. And what I would like to show you are actually clustered results from 3 different cell types. Now, this is the same T-SNE plot that I've used all along, but I've grayed out the areas that we're not going to speak about in favor of the 3 different epithelia that we will speak about. Now, the first 2 are probably the most obvious ones. Those are your luminal epithelia. You can see many, many cyan dots overlaid with the DAPI signal. Each one of these is presumed to be a luminal cell. There's 32,383 of these in this image. And then in dark blue, you can see dark blue regions here, where the collecting duct is. There's one up here. There are definitely not that many, 546. It's pretty clear that they're around. But then the result that I found very fascinating, and the one I will spend the remainder of our time on, is this luminal keratin 8, keratin 14 cell. There are only 44 of these cells in this entire image. So that's less than 1 percentage point of the entire cell population. So the question is this noise? Why would he dwell on this? It's only 44 cells. Well, that was my initial question as well. It's like, what is this? Why is this noise here? How can I get rid of this? Fact is, I could not remove these data from my clustering algorithm. It didn't matter how much I constrained the parameters. Somehow, these cells always showed up in my output. I didn't ask to see these cells. I don't know what these cells are. They just presented themselves to me. I was following the data, and here we have a tiny little population of 44 cells that I spend a lot of time looking at now. So let's look at these cells. This here is, again, the same representation, the same T-SNE plot. And what you can see up here is keratin 8, shows you cyan, your luminal cells, keratin 14, blue, your basal cells. And then in here, now you will understand why these are called keratin 8 and keratin 14 cells. You can see the cell body and a couple more up here. Those are the luminal keratin 8 and keratin 14 cells. Now, if I would look at just this image, I would probably dismiss it because it is actually, by my admission, ambiguous. Maybe this has just been overexposed. Maybe the keratin 14 marker is just so strong that it washes over and these cells just happen to be labeled. Maybe it's the same as down here where you can see really nice basal layer and you also have cyan cells. So what's different between this and that? Well, this is where the CODEX system becomes in really handy and where it's very, very powerful to know that you have additional biomarkers. So this heat map shows you our luminal cells, cyan, the keratin 8 and 14 cell in orange, and the basal cell in blue. And it shows you 36 biomarker expression patterns normalized, where 1 is the maximum, or red. And you can see there aren't a lot of immune cell markers, but our epithelial markers certainly have a nice spotty expression pattern that we can dive into a little bit further. So let's take a look, for example, at TP63, down here on the bottom right side. TP63 is a key marker for basal cells. So if you look down here, this is sort of your canonical basal and then your secretory epithelia. Basal cells have this really nice TP63 labeling. See these nice slender nuclei. Now let's go up here where our supposed keratin 8, 14 cells are. You can see that the TP63 very, very selectively labels only the outer layer, the basal cells. There's still a group of cells on the inside that is not labeled. There's these guys in orange. Let's look a little bit further. Look at our heat map. Beta-catenin, you can see here, should be shown in blue. Labels these keratin 8, 14 cells, but not the other ones so strongly. Well, yes, indeed. They are showing up in blue, which is beta-catenin, whereas the basal cells don't seem to be, most certainly are not down here. So this anatomically and by virtue of biomarkers discriminates these cells from all the other ones that we see in this image. Then finally, TFAM. You can see that these cells are very strongly TFAM expressing. Other cells do express TFAM, as you can see in your heat map, but not nearly as much. So this is one of the things that I think is most exciting about CODEX. And that is this toggle between big data, or let's just say T-SNE plots, something that single cell biology is, heat maps, and actual hard anatomical data at single cell spatial resolution. All of these windows can be opened simultaneously in your software if you want, and you can toggle back and forth. You can click, unclick any marker you want here, and you can ask whatever question you want at whatever zoom value. Remember, we're just looking at a fraction of this entire tissue piece, and we're really only looking at 4 or 5 markers right now. This tissue is another 20 times larger, and there are 36 markers. So there are many, many, many questions to be asked and answered. Now, I would like to conclude this part here by just really bringing home the point of what we've just done and whether or not this really is reliable. So we've just talked about the keratin 8, keratin 14 cell type that are distinct from TP63 basal cells, but yet they comprise only 0.07% of the image population. It's very, very small, little tiny group of cells that maybe we're spending too much time talking about them, but that's not the question. The question is, are these data really reliable? And what we can show you to answer this question is another result from the same paper in Nature Communications by Nguyen et al. That shows exactly this cell type. So you have keratin 8, you have keratin 14. You can see them here, and they overlap. This cell is the same as the cell type that I've just shown you. Now, the authors of this paper, by my own admission, they probably know many more times about breast tissue than I do. They have said that this is the first description, theirs, of this cell type in humans. It was known prior to their experiment that these cells exist in mice, but not in humans. This is normal breast tissue. To our knowledge, this is the first demonstration of this cell type in human cancerous tissue. We have not seen another paper that has described these cells in human cancerous tissue. So keratin 8, keratin 14 cells are present, but extremely rare in normal and in tumor breast tissue. So we have just made a discovery. We have used a CODEX system in FFPE tissue with high-plex biomarker analysis to make this discovery. So that being said, I would now like to summarize what I've just told you and offer you a little bit of perspective. So we talked about single cell biology in FFPE tissue. I hope I presented to you that CODEX is a very nice tool that exists in a space between, let's say, techniques like single cell transcriptome sequencing and conventional clinical histopathology. CODEX is in between. It allows you to take the best from both of these worlds and combine them into one. And very, very importantly, it allows you to get very precise and unbiased cell phenotyping. Every result that I've shown you today recapitulated another result elsewhere. This was done by accident. We didn't plan to do that, but the results are strikingly similar. Every result that I've gotten here, or that I've gotten to today, was reached after unbiased data analysis. I had no preconceived ideas about this tissue. I just let the data guide me to where I am today. Finally, and probably most importantly, single cell spatial resolution was maintained throughout this entire journey. We started with a tissue, and that tissue still is present. We have the information about every cell and where it is. Now, I didn't show you any of the algorithms that we have to analyze spatial interactions. That's a topic for another discussion. But what I've shown you is that single cell biology is possible to do in situ in FFPE tissues. And that is really important, because if you take this into perspective, there are about 120 nationally recognized biobanks worldwide. Some of these biobanks have incredibly rich repositories of individual tissues. For example, the thyroid biobank at Chernobyl has up to 5,000 pieces of thyroid tissue. Imagine what you can learn from analyzing 5,000 thyroid pieces with 36 biomarkers. It's hard to imagine because there's a lot to be learned. FFPE preservation to this day remains one of the key or one of the most widespread preservation methods in biobanks. It's cheap, it's robust, and it's widely available. So this technique will not go out of style. There will always be FFPE tissue, and there's so much of it already just waiting to be analyzed. So at this point, I hope that a lot of you are convinced that it is possible to unlock discoveries with CODEX anywhere at any time in a very high throughput, very efficient, and also very economical manner. So with that, I would like to thank you very much for watching, and I would really like to give acknowledgement to my coworker Nadia Nikolina, who's done a beautiful job on these experiments, worked really hard to come up with this. This is her baby, and it's a fantastic result. It was a great pleasure to analyze this. To OneMean and to Andy, who have helped along this process as well. So thank you very much for watching.